welcome. We're going to begin our next session. I just ask that everyone please be seated, get settled. I know everyone's excited for lunch. Now we're just going to come right back in. And we have an exciting set of discussions for you. My name is Jody Ann McLegan. I'm from Columbia University, the Coordinating Center. I work very closely with George, Patrick, and the rest of the Columbia team. We are happy to have you here. So for today's session, we have an exciting group of panelists that includes representatives from around the world. They represent our US, APAC, and or European chapters. They are joined by our regulatory agency representatives, and they will discuss OHDSI's ongoing support for regulatory agencies and their regulatory decision-making using real-world evidence. You will hear about the challenges and questions that these agencies are thinking about and ways in which OHDSI is working to innovate, provide support, and meaningful long-term solutions. First, we're going to hear from the agency representatives, followed by the presenters. Each representative will discuss their specific need or challenge, and then they're going to introduce the OHDSI representative that is working on those needs and challenges. I am particularly looking forward to hearing all of your presentations today. Doctors Fan Bu, Seng Chan You, and Peter Rijnbeek. Fan specifically is looking from University of California, will be presenting the work that she has been leading with members of the US FDA CBER team to examine and improve vaccine safety surveillance methods and develop more flexible approaches. Seng Chan You from Yonsei University will discuss the work that he has been doing to support the Korean FDA administration on their work in replicating clinical trials using Korean databases, and with the specific goal of determining whether or not they're able to achieve the same effect estimates as those observed in clinical trials. So I ask that you take a moment. Before that, though, we'll also hear from Peter from Erasmus MC University Medical Center of Rotterdam, which is the central coordinating site of the DARWIN-EU initiative. And he will be discussing the support and the work that he's been doing there. This is a very large initiative, and I'm very excited to hear about that. So we ask that you take a moment, mute your phones, everyone, listen keenly, and I task you with thinking critically about the challenges that you will hear today, and also think about the potential solutions that we can continue to come up with. So with that being said, I'm gonna first introduce Dr. Patricia Lloyd, who's a statistician. She is with the FDA CBER team, and she works her and her team work collaboratively with the OHDSI community to improve vaccine safety surveillance method. And it's such a pleasure to have you here today. Please help me welcome back Dr. Lloyd to the stage.
Thank you, Jody. And hello, everyone. Post-market safety surveillance for new vaccine products is essential for detecting rare or high-risk adverse events that may not be discovered in clinical trials due to limited sample size. At the FDA, such surveillance is conducted sequentially by analyzing streams of observational data, such as spontaneous reports, insurance claims, and EHRs. The major challenge in repeatedly analyzing accruing data is that the need to control the sequential testing error while being able to signal rapidly. The current standard statistical approach for this type of surveillance is max SPRT, which is designed to handle sequential multiplicity. However, the drawback is that it requires pre-specifying the entire surveillance schedule. This is inflexible and inconvenient, as oftentimes we wish to adapt or extend analysis timelines given the data situations. Therefore, we hope to develop more flexible methods that do not require a prefixed schedule. At the same time, we want a method that corrects for inflated false positive rates induced by systematic error in observational data, which is a prevalent issue in the practice. So together with OHDSI Methods Research Group, we have been developing a Bayesian framework that has these desired features. Today, a member of the OHDSI team, Dr. Fan Bu, will discuss this Bayesian framework and present some very promising preliminary results. Thank you.
Thank you, Dr. Lloyd, for the introduction. As Dr. Lloyd already mentioned, the goal of safety surveillance is to detect increased risks of adverse events after exposure to vaccines. Statistically, this is a hypothesis testing problem. The null hypothesis is that there's no increased risk, so we say there's no safety signal. And the alternative hypothesis is that there is an increased risk, so we say there is a safety signal. The challenge here is that we want to be able to make decisions fast, so we have to analyze data on the fly as streaming data become available to us in monthly or quarterly batches. And because testing errors can accumulate as we repeatedly analyze data, this is a really difficult problem. Nowadays, the current standard approach to do this is to use a frequentist approach, the maximum sequential probability ratio test, MAX-SPRT, coupled with historical rates comparison design. Roughly speaking, it works in this way. As data become available, we can sequentially compute the incidence rate of adverse events over time, and then we can compare it against the historical rate computed from historical data. And then if the rate ratio is significantly above 1, then we can say there is an increased risk. Now, we'll have to do this in a sequential testing manner. So MAX-SPRT, given a specified alpha level, say 5%, and given a specified surveillance schedule, say 12 months of analysis, can compute a threshold for the log likelihood ratio statistic. Now, over time, as data become available, we can compute the log likelihood ratio statistic. If it exceeds the threshold, then we can stop the study, reject the null, and declare a safety signal. This is relatively straightforward, but as a widely used method, MAX-SPRT does come with problems. The first major issue is that we have to pre-specify a surveillance schedule, including the total length of analysis and the expected sample sizes of data that are available in each observational interval. And we have to stay fully committed to the schedule, meaning that if things change in real life, as they always do, we cannot adaptively adjust our analysis plan. And that's hugely, hugely inconvenient in practice. The second, perhaps more severe issue is about the systematic errors or biases in observational databases, which are the data we have to work with in surveillance. And the problem with systematic error is that it can hugely inflate the type 1 error rate in our studies. So oftentimes we would operate under the alpha level of 5%, so we think that the false positive rate is 5%, but because of the presence of bias, the empirical actual type 1 error rate can be much higher. And we have seen that sometimes it can be more than 5 times higher than the specified alpha level. And that's really concerning because we would be making a lot of bad decisions by producing a lot of false positive signals. And what's even worse is that we would not even realize that we were making those access mistakes because we are ignoring the impact of bias. We would think that alpha is 5%, but actually it should be much higher. And so being able to handle bias is really important, but under the framework of MAX-SPRT, it's not easy to do it because it's not easy to develop a statistically coherent and principled approach. Because of those reasons, we want to develop a better alternative approach under a Bayesian framework, which I will talk more a little bit later. And in order to benchmark the performance of our method against MAX-SPRT, we have done some empirical evaluations using real-world healthcare databases. So basically, we would do some retrospective analysis of data by simulating the data streaming process in monthly batches. We have been using historical vaccines because we know their behaviors better compared to newer vaccines such as COVID-19 vaccines. And we have been using a large set of negative and positive control outcomes for which we know the ground truth values of their rate ratios so that we can empirically evaluate the false positive and false negative errors. And I will present some representative empirical results just after I talk about the details of our method. So Bayesian inference is different from frequentist approaches in that it can sequentially update our knowledge about the data evidence by sequentially producing something called the posterior distribution for the rate ratio of interest. So usually in analysis at earlier stages, if you look to the left side, the posterior distribution would be rather flat, indicating a high uncertainty about the actual value of the rate ratio because there's little information available from data. But if you look on the right side, at later stages of the analysis, this posterior distribution will become more and more concentrated towards the value indicated by the data. And that's when we have more data evidence. That's when we are more confident and more certain about the value of the rate ratio parameter. So what's really nice about Bayesian inference is that it not only just offers a bunch of point estimates, as in frequentist approaches, it gives us this distributional characterization that reflects the amount of information from data and our uncertainty and certainty about what's going on. So something that can easily come out of the posterior distributions is the posterior probability for a particular hypothesis. For example, here on the bottom, the blue line is the posterior probability for the alternative hypothesis given the data at any time point. And this value is truly a data-driven quantity that tells us how much we should believe in a particular hypothesis given the data that we've seen so far. And what's even nicer is that we can directly use this probability quantity as the test statistic in our sequential analysis for surveillance. For example, here, this red dashed line can be an assumed decision threshold. For example, here it's 95%. And we can sequentially compare the posterior probability against this threshold. And when the threshold is crossed, for example, here at month 10 in this example analysis, that's when we can stop the study and say that there is a safety signal. So on top of establishing this really straightforward, highly interpretable process for sequential analysis for surveillance, we have also proposed a joint statistical model to correct for the bias in observational databases using negative control outcomes. So Martijn already talked about negative control outcomes. Basically, a negative control outcome is an outcome that has no causal relation to the exposure of interest. So the estimate for the rate ratio should be very close to 1 if there's no or little residual systematic bias in data. However, if the estimate is away from 1, then that estimate can also tell us something about the scale of the amount of bias in data. So using that logic, we can do something like this. First of all, we can produce a posterior distribution for the rate ratio of interest. Here, everything is in log scale, so a log rate ratio of 0 is the same as a rate ratio of 1. We would know that the posterior distribution would probably be biased because of the systematic error issue prevalent in databases. At the same time, we can also estimate a distribution to describe the amount of bias in the data, in the analysis, by analyzing the negative control outcomes. And then we can jointly model those distributions to produce a de-biased version of the posterior distribution for the rate ratio of interest. And we can do this at every time step of the analysis to adaptively learn about the bias and adaptively correct for bias. So I'm going to present a little bit of our empirical results. And in our results, we have results for the full Bayesian approach with bias correction, but we also have some results as a baseline for the more vanilla version of Bayesian inference without the bias correction component so that we can also get a sense of the impact of bias correction. So the first set of results I want to present is about the type 1 error rates over time, over the full span of the analysis. And those are empirically validated using the negative control outcomes in our study. If we look at the first curve here on the top, that is the type 1 error rates of max SPRT. This max SPRT method actually operates under the alpha level of 0.05. But if we look at the number there, the actual empirical type 1 error rate is as high as 27%. So that's 5 times more than 5%. This is really a bad thing because this could be already happening in a lot of surveillance studies. By using mass SPRT without doing something about the bias, this is what we can actually have. We will be making a lot of false positive decisions without even realizing that we are making those mistakes. And if we look at the bottom curve here, right on the bottom, that's the performance of our Bayesian procedure with bias correction. Very interestingly, we, by adjusting for bias, can restore the empirical type 1 error rate level to about 5%, which is oftentimes the desired level. I say interestingly because our Bayesian procedure targets the estimation bias, not actually any specific alpha level. But by being able to acknowledge bias and handling bias, we can remedy the issue of type 1 error inflation, and that's the kind of important message we try to convey here. The second set of results is about statistical power in terms of how precisely we can identify a safety signal if there's actually a safety signal. Those are evaluated using positive control outcomes, and those are also evaluated over time. So here, in order to make fair comparisons, we have chosen Bayesian decision thresholds to make sure all the methods have the same level of type 1 error rates empirically. So if we look at the leftmost side, that's the max SPRT power, and the rightmost side, that's the full Bayesian method with bias correction. And if we actually look at them and contrast them, we can see that the Bayesian method is consistently outperforming max SPRT in terms of statistical power. And of course, I'm only showing one example analysis, but this is pretty much what we are seeing across the board. In many other analysis examples, the Bayesian method would at least have the same amount of power, if not higher, compared to max SPRT. We do want to emphasize that there is intrinsically a trade-off between controlling alpha level and improving statistical power. In the context of safety surveillance, which is mostly a tool for initial screening, perhaps high sensitivity is what we truly want because we do not want to miss any important signals. By using a method like mass SPRT without adjusting for bias, we could potentially have high sensitivity, but the price we have to pay is a hugely misspecified alpha level, a hugely misspecified type 1 error rate level. And that's really bad because in practice, we may get an actual type 1 error rate level that's much, much higher than 5%. So we'll be making a lot more mistakes. We'll be making a lot of bad calls. What we want to advocate for is to use a better statistical method, such as ours, that is more correct, more coherent, and more principled. If high sensitivity is truly what we want, then perhaps we can just use a larger alpha level instead of 5%, which was pretty much arbitrarily chosen by Fisher many decades ago. We can use, say, a level of 10% or 20%, and then use a good statistical method that can stay true to those pre-specified error levels and be very transparent about those operating characteristics. We believe that this is the way to go moving forward if we want to have more reliable, data-driven decision-making in healthcare. So to summarize, we have developed a Bayesian framework for safety surveillance that is more flexible, less biased, at least equally powerful, if not more powerful, compared to the current standard approach, max SPRT. Of course, on our side, there's still more work to be done. For example, currently we are exploring a double threshold strategy with an additional lower bound that can allow us to stop the study early if there's sufficient amount of evidence in support of the null hypothesis. hypothesis. By doing so, potentially we can save more time and more resources in safety surveillance. I do want to say that this is really a team effort, so I want to acknowledge all the team members who have been working together for their individual inputs and contributions that made this work possible. And of course, we're very grateful to the FDA CBER Center for their continued guidance and support throughout this project. And finally, if you want to learn more about our work, you can reach out to Mark or come talk to me or the FDA representatives at the exhibitor table, which is down the hallway on the other side of this hall. Thank you so much. That's all I have to say.
Thank you, Fa, for that wonderful presentation. Next up, we're going to have Seng Chan You, and Dr. Yu will be introduced virtually by the Korean Institute of Food and Drug Safety Evaluation representative, Yeon-Hyu Lee.
Hello, I'm Yeon-Hyu Lee, a senior scientific officer at the Ministry of Food and Drug Safety, Korea. First of all, I would like to thank OHDSI Symposium for taking interest in MFDS Korea research project on real-world data and providing the opportunity to present at the symposium. Please excuse not being able to present in person due to COVID-19. I will proceed to present in video format. Today, I would like to briefly introduce you to the background behind MFDS Korea's real-world data research project, our progress thus far, and the plans moving forward. Randomized controlled trials are the gold standard for evaluating the efficacy and safety of drugs, but they apply string inclusion criteria, which makes it difficult to apply the result in the actual clinical setting. There's an increasing need for clinical research using medical big data. When developing drugs that come with ethical or realistic hurdles to conducting clinical trials. Also, in post-marketing safety surveillance, the need for the use of real-world data generated at the healthcare centers, such as claim data and medical record, is growing, in order to move beyond the limitations of existing drug safety measures built in user-resolved surveillance. So MFDS Korea announced that post-marketing safety surveillance can be conducted using medical big data last year. Our department, in preparation for the growing demand for the use of real-world data in drug development and safety management, has formulated a research project to introduce and utilize real-world data throughout the drug life cycle for drug development, and have continued our efforts starting in 2020. In order to use real-world data for drug approval process and safety management, we surveyed related Korean and international guidelines and cases, and conducted pre-research to share diverse opinions and increase accessibility to Korean medical data. In addition, we are supporting replication studies using real-world data for pivotal clinical trials to establish drug safety surveillance system based on real-world data. Through large-scale funded observational study using big data, we evaluate the quality and reliability of Korean real-world data and problems in real-world data analysis to derive limitations and considerations on research design and analysis, and prepare Korean applications and improvement plans to build a systematic basis to utilize real-world data. Looking into the future, in order to prepare a method to verify the quality of the real-world data, we plan to support research on data quality management and standardization for regulatory decision-making. We also plan to support research to identify status of Korean registries, registry management, and the data utilization method. Integrating and analyzing all of these results, we will present standards to strengthen the reliability and accuracy of medical big data analysis, and support efficient clinical research projects. Taking advantage of the fact that Korea has a nationwide administrative claim data enrolling whole Korean population and nationwide CDM network, we will continue to promote real-world evidence-related research in Korea. If you have any question related to presentation, please reach me through Dr. Chan. Thank you for your attention.
So I'd like to argue why we need evolution of evidence-based medicine and why we are applying clinical trials using EHRs. I'm Seng Chan You from Yonsei University College of Medicine, Department of Biomedical Systems Informatics. This is CY of mine, and this research was supported by a grant from Ministry of Food and Drug Safety in Korea. Current paradigm of evidence-based medicine define that usual randomized clinical trials provide high quality of evidence, whereas the usual observation study provide low quality of evidence. Even though the Martin and Patrick show how we can provide high quality of evidence by using observation study in the previous sessions. The strengths of the randomized clinical trials is based on the magic of randomization. The magic of randomization guarantees the unbiased distribution of confounders, which results in the high internal validity. However, this high internal validity does not guarantee the generalizability to different or real-world population, because the real-world population has much more diverse. So I believe that in the most cases, the trials cannot enroll more than 0.001% of the patients with the indication for that intervention, because among the various steps for the clinical trials. So to judge whether the results from the trials can be applicable to the patients in front of us, we need to clearly understand the eligibility criteria. And after the publication of pivotal clinical trials at the symposiums, we usually heavily discuss whether this clinical trials results can be applicable. And at the symposium, we ask, to whom do the results of this trial apply? Can the results be reasonably applied to the definable group of patients in a particular clinical setting in routine clinical practice? Are the results from the clinical trials generalizable beyond the trial setting? Until now, the external validity of RCTs has largely neglected. So rules governing the performance of trials do not cover issues of external validity. Drug licensing bodies such as US FDA do not require evidence how much the trial population is representative of routine clinical practice. None of the many scores for judging the quality of clinical trials address external validity adequately, and there are no accepted guidelines on how external validity of clinical trials should be assessed. Previously, OHDSI studies shows somewhat different results from the clinical trials. The Plato trial, which was published in the New England Journal of Medicine in 2009, showed that ticagrelor had better efficacy compared with clopidogrel in patients with acute coronary syndrome. In a decade later, we showed that the beneficial effect of ticagrelor was not evident in real-world population. Similarly, the accomplished trial showed that the ACE inhibitor plus calcium channel blocker is better than ACE inhibitor plus thiazide in high-risk hypertensive patients, which was published in New England Journal of Medicine in 2008. A decade later, we show that there is no difference in the effectiveness of these two combinations in hypertensive patients. Why this difference happens? Dr. Everett and the Columbia team published a paper entitled "Eligibility Criteria Fail to Eliminate Clinically Significant Differences Between Real-World and Study Populations." They found the substantial heterogeneity between the trial enrolled patients compared with the Columbia University's real-world population. the percent of patients with older than 65 years was 66% in the accomplished trial, but it was less than 20% in the patients with indication and use those combination in Colombia, whereas the patients with use those drugs but meeting the eligibility criteria has a much higher age distribution. But you can see that only 4,000 patients met eligibility criteria among the patients with indication for that drugs. These patterns were found in the emulation of other trials. How this difference in baseline characteristics can affect on the average treatment effect. Depth and extent depth study can be exemplary. The depth study published in the New England Journal of Medicine in 2014 showed that the 30 months of dual antiplatelet therapy can decrease the risk of major adverse cardiovascular and cerebrovascular events compared with the 12-month standard depth therapy. Later, the Butala and Dr. Ye's group showed that conducted extent depth study. In the extent depth study, they re-weighted the trial population to be similar to the current clinical registry's patients. In these re-weighted patients, the benefit observed from the clinical trials diminished. So there is no benefit anymore in this re-weighted patients, similar to the current patients in the registry. I and Dr. Kromos wrote the editorial to this paper, and the title was The Evolution of Evidence-Based Medicine. And we asked, why are we left with leveraging real-world data to address the generalizability of the results from the clinical trials? The reason why is that the characteristics of enrolled patients meeting eligibility criteria in the trial may differ from the patients under routine clinical practice. And moreover, over time, the characteristics of people of indication have changed. The evidence from trials may not be durable over time and may not be durable after decades. So that is why we initiated the TRIAL project, trials replication through observation study by Yonsei. In this project, we are trying to emulate 15 target trials to identify difference in baseline characteristics and to estimate heterogeneous treatment effect. The trials we are emulating are targeting GLP-1 agonists, SGLT2 inhibitors, DPP-4 inhibitors, prasugrel, ticagrelor, NOACs, and tofacitinib. There are two cohorts we are creating in the TROY project. The first one is the eligibility criteria cohort, in which we are trying to replicate the inclusion and exclusion criteria as closely as possible to the targeting RCTs. We found that the eligibility criteria in the trial are really complex, and it was not really easy to implement in this study. The other cohort is indication-only cohort. In this cohort, the patients were enrolled when they used the drugs and then with the indication according to FDA label. And we estimate the differences between the eligibility criteria cohorts and the inclusion-only cohorts with the RCT enrolled patients. And all source code for this work are available at GitHub. Until now, we do have results from the two data partners, as University of Health System, Severance Hospital, and Aju University School of Medicine. In this table, on the right two columns show that how much the eligibility criteria covers compared with the indication-only cohorts. The green color means that more than 30% of patients of indication can be covered by eligibility criteria. And yellow means that it covers less than 30%, but more than 10%. And red means less than 10% can be covered by eligibility criteria. You can see that the trials, that when we apply the eligibility criteria for the trials for anti-diabetics that usually no more than 30% of the patients were covered by eligibility criteria. In other words, it means that the FDA label is much broader than the eligibility criteria of trials, which is similar in the trials for antiplatelet and NOACs. And there is substantial heterogeneity between trial population and the real-world population. You can see that in the Plato trial, the median body weight was 80 kilograms in the patients, but in Korean real-world patients, the median body weight was only 65 kilograms. And there is more comorbidities in the RCT enrolled patients compared with the real-world patients. And when we compare the indication-only cohort with the eligibility criteria cohort, then the cohort with eligibility criteria had more comorbidities when we emulating the trials targeting apixaban, edoxaban, rivaroxaban. And these findings were similar, and when we emulate the trials targeting SGL2 inhibitors, and DPP4 inhibitors. So in conclusion, the results from the clinical trials do not actually guarantee the external validity in contemporary routine clinical practice. And our results reveal clinical difference between the population enrolled in RCT and the population replicated from the observational databases. Once again, these findings emphasize the need for examining evidence using real-world data to generalize the evidence from RCTs. Thank you for your attention.
Thank you, Chan. Next, we'll hear from Peter Rijnbeek from so he will be talking about the DARWIN-EU initiative. And he is introduced by Amir Cook, the Executive Director of the EMA and the co-chair of DARWIN-EU Advisory Board.
DARWIN-EU will provide EMA's scientific committees with real-world evidence from healthcare databases across the EU, and it will be central to prepare for and respond to future public health crises such as COVID-19. So what could be more timely? The vision of DARWIN-EU is to give EMA and national competent authorities access to valid and trustworthy real-world evidence, for example, on diseases, patient populations, and the use, safe, and effectiveness of medicines, including vaccines, throughout the life cycle of a medicinal product. By supporting decision-making on the development, authorization, and surveillance of medicines, a wide range of stakeholders will benefit, from patients and healthcare professionals to health technology assessment bodies, and, of course, the pharmaceutical industry. DARWIN-EU will act as a pathfinder for the European health data space and will ultimately connect to the European health services, enabling the use of this data in the context of medicines regulation in Europe. EMA will be working with the Erasmus University Medical Centre Rotterdam to establish the DARWIN-EU Coordination Centre, and you will hear a lot more about this today, because the Coordination Centre is designed to develop and manage a network of real-world healthcare data across the EU, and will be able to conduct scientific studies requested by medicines regulators and at a later stage requested by other stakeholders. The contract was awarded following an open call for tender, and we're delighted to have Erasmus Medical Centre as the contractor, because the medical centre is highly experienced in database management, in federated analysis and multi-database studies, as well as being in a position to use a common data model, and which means that this can be readily available to a large number of potential data partners. We will work collaboratively to ensure that DARWIN-EU rapidly delivers benefits, and we will pilot regulatory use cases across the life cycle of medicines.
Yeah, yes. You can imagine that we are pretty proud that we got this video. And I must say, this is also a thank you to the whole OHDSI community. I think this is not something that only Erasmus is delivering, but it's something that we could do because we are part of the OHDSI community, and all of you have done a great job. And this is, of course, for us, really the cherry on the cake to start delivering real-world evidence to the regulator in Europe. So it's very exciting. And it's especially exciting because in Europe we have a lot of challenges that you are all familiar with. Different types of structures of databases, but also the problem of a huge number of terminology systems that we have to deal with in Europe. So I might even say that our approach with OHDSI is even more applicable to the European situation, where we have to solve all these problems at a very large scale. We don't speak one language in Europe, we have many different solutions, and we need to find methods to solve that problem, definitely if we want to scale up to a very large number of studies. And that's where DARWIN-EU has been, and that's why it has been initiated. So the vision of DARWIN-EU is to establish and maintain a framework supporting better decision-making throughout the life cycle of the medicinal products with timely, valid and reliable evidence from real-world healthcare. And the objective is to establish and maintain a continually enlarging network of accessible observational data sources standardized to the Open Common Data Model, and to execute all steps of high-quality non-interventional studies within the network, and then to make these results also available to the European Medicines Agency and their committees. To do this, and especially at the scale that EMA wants to do this, we have to really generate or implement a paradigm shift. We cannot simply add more teams that run more studies in parallel. We have to be much smarter, and that's where, of course, everything that we've been working on in the community is stepping in. We need to standardize things. We need to standardize the data. We need to standardize the analytics. But we also need to standardize the reports. We need to agree on what we mean with the drug utilization study and not invent the wheel over and over again. So it's not simply scaling up traditional approaches. DARWIN-EU is going to be, of course, a federated network where the data stays local, standardized to OMOP, and we bring the tools to the data. And that's especially important in Europe, where we have the GDPR regulation, all about privacy. And this is a very good solution that people like because of that. So the coordination center is going to be in the middle of this diagram. There's the EMA on one side asking for questions, and on the other side are the data partners, either directly interacting with the coordination center, but there's also some thoughts about data permit authorities in Europe, where, for example, a country is represented by a data permit authority and studies would run through that organization. This is part of the European health data space development as well, and it's not completely clear how this is going to work, but we'll definitely have experience with that at a later point in time. So setting up the Darwin Coordination Center, as was already said earlier in the video, was done through a tender process where we were very, very happy that we were selected to set up the Coordination Center for the EMA. And that's not something we do alone. We work together with very good subcontractors that we have worked with before, and you probably are all familiar with definitely University of Oxford with Dani. Synapse may be new to you, but it's a management organization that we have worked with many, many times in large European projects, and they are fantastic. IQVIA, you all know, the Hive, very experienced in mapping data, and much more. And Odysseus, you all seen as well here, and also have a stand, who are building very interesting tools for us to use, like Arachne and other tools. But also are, of course, very involved in the vocabulary, and that's going to be extremely important for us. I'm supported by Dani and Katja, and I think the three of us together with this larger team are going to do a great job, I'm sure. The establishment is built around this kind of structure, so there's an executive board, the director and the deputy directors. They're supported by advisory boards that we are currently setting up, and we are selecting different types of experts to join these boards. And under that, under the director, are the 4 different pillars. One is development, where we are building all the tools, the analytical tools, but also things like templates for protocols, templates for reports, etc. Then there's operations, and operations is composed of 2 different parts. One is study operations. That part is going to be responsible for the execution of all the studies, setting up the teams, doing the dissemination. And the other part is network operation, which is all about standardizing the data to OMOP, if it's not done already, or otherwise do quality control checks on the already mapped data. Then there's a technology pillar where we are working on adding additional tools in the ecosystem, but also supportive tools like a service desk. We're setting up collaborative environments to work in. We are working setting up a remote research environment, which is a secured environment where the data partners could upload their results to, and we can lock down that environment to actually execute the study, or to look at the results of the study, I should say. And of course, training platform, where we will be building on the great efforts that has been done in Eden, like the Eden Academy, and other training courses that are available through OHDSI, because doing the change management in such an organization is also a very important part of our work. We have to train the people to use this new type of processes and results. And then there's a management section which deals with a lot of things like conflict of interest, which is of course very important. But also, I can tell you, we are producing a huge amount of deliverables. I think we've done a world record on the number of deliverables that we have written in the last couple of months. So thanks a lot for the management team. Now, what's EMA's role in this? Clearly, they are going to be the ones that are asking the questions coming from their other committees. But they're also highly involved in setting up an establishment and in the establishment phase of the DARWIN-EU Coordination Center, which is fantastic. So we work with a very nice team on the EMA side, where we have, in principle, twice weekly meetings where we discuss the progress and discuss all type of procedures and processes together with them to get alignment. And I really appreciate that interaction. So the data sources that we are going to include are the ones that you would expect. So the diverse set, nice geographic spread, and it's a growing list of data sources over time. I'll talk a bit more about it in a second. Oh, actually this one. So it's growing up to 40 databases in the first 4 years, but it may be extended further after that. The process that we have been developing, I will not go through the whole slide, but I just want to mention that it's very important that we go to a quality control mechanism with the data sources. So even though we ask them for their interest, we have meetings with them to align on what we actually expect from the data partners. We also go through a process where, beside them filling in like a questionnaire with all important questions on how often are you actually updating your data source and what are your governance procedures. We also ask them to run a package against their CDM that generates all kind of information on the trends of the data. Also, we check like the speed of queries on their infrastructure to make sure that we will not encounter these type of problems later on. So this tool that's built is based on the tool built in the Eden project that we use it there to onboard data partners in the large data network of Eden. We extended it a little bit here and now automatically generate an annex to the onboarding documents, and that can be sent together with all the information to the EMA for approval, and after that we sign the contracts with the data partners and then have a contract per study, so a framework contract and a work order. The type of studies that we're running in Darwin are what they call off-the-shelf, so quick studies. You can think about incidence rate studies or drug utilization studies, more complex studies, safety, for example. And then there's the option for them to repeat those studies, so run them on a quarterly basis and get updated reports. And then finally, there is the idea of very complex studies. It's not completely defined yet what that's going to be, but it's assumed that that may require much more custom programming, while the other two should actually be based on standard analytics as much as possible. So the process is going to be EMA's requesting, has a question. We look at the feasibility of the study, which is a topic by itself, to think how you actually do a good feasibility study across a data network. And some of the tools that we have seen today, like the data diagnostics, for example, could really help with that. We then have protocols based on standard templates, so we're not going to write it from scratch all the time. It's been shared with the data partner together with the code. They run it. It's been reviewed by the Coordination Center, sent back to EMA, built in a aggregated format that graphs across multiple databases. And they review it and then make their regulatory decision making. Now, the number of studies is really ambitious, so it's growing very fast. So we're now in the first phase of the project, year 1, I think month 7 or so. This year we only have to run 4 studies. But it's growing fast, so next year it's already 16 studies, and it's going up and up and up to almost 145 studies a year. So that is a huge number of studies. That's not possible if you would need all kind of teams to run that. You need to be smarter, and we are investing a lot of effort in figuring out how to do that and how to scale up. Of course, that's done through standardized analytics, as we all know. We just need to change the drug of interest and not build a whole package again to run a drug utilization study. It's a very interesting thing to work on, I think, at the moment, because we have to align on what we mean with all these things. What is a drug utilization study? What is the report that we need to generate? And alignment is being sought now with EMA. Currently, we're working on these type of analytical pipelines or use them from OHDSI. Population-level disease epidemiology, patient-level disease epidemiology, population-level drug utilization, and patient-level drug utilization. And you see some examples of the studies that may fall under these categories on the right. More complex studies are risk minimization, effectiveness, new user active comparators studies, where, of course, the cohort method and other nice tools from OHDSI would be used. And self-controlled case series is another topic that's on the list now. So where are we? So we are now in month 6. We've done a lot of preparatory work. Also started to build some new pipelines. The processes are all in place. We are actually onboarding data partners already. 10 data partners have been selected by EMA, and the first are actually already signed up and we have contracts with, which is great. And other tools are being developed in the development pillar, and a lot of templates and work there. Operations, we have been requested 3 studies, and these studies are, of course, drive our development, characterization study, drug utilization study. And on technology side, there's a lot of progress, and I'm really looking forward to share some of that also in upcoming OHDSI Symposia. And finally, we are also moving to phase 2. So this is a contract that's every year has to be extended, and this is now also being approved. So I'm very happy that we are able to continue, of course, and deliver these studies at a large scale to the European Medicines Agency. If you have any other questions, I will be at the booth together with the FDA, and you can definitely ask me a lot of things about DARWIN-EU. And otherwise, there are some links here, and there's also a website coming up from DARWIN-EU where you will find much more information. Thank you very much.
Thank you, Peter. It's great to hear about that large undertaking. And I look forward, and I'm pretty sure the rest of the community does as well, looking forward to hearing more updates about the progress and the results that comes out of that. So I know we're right on time. So 2 things I'm going to ask you all to do. I know many of you have questions for our panelists. They're going to be available to take any questions that you all have by the FDA exhibitor table, which is right outside of the ballroom to the right of the poster presentations and the demonstrations that are going on. And secondly, following the sessions, we have our poster presentations, we have poster walkers to guide you all along. So please, anyone that's leading the poster walk, if you could just raise your hand, raise your sign, and we encourage everyone to go there, take a look at the posters. We have a lot of great demonstrations, posters for you all to view, so please do so. Thank you.